Hey everyone, ready to dive into some really cool science with us today?
Always up for that. What are we diving into today?
We're talking about iPSCs.
Induced pluripotent stem cells. Definitely a game changer, especially for neurological diseases. It's a field exploding with potential.
It is. Neurological diseases, things like Alzheimer's, Parkinson's, they present a huge challenge, don't they? Millions of people.
Absolutely. Millions affected. And treatment options, well, they're limited. That's what makes these iPSCs so fascinating. Huge potential for new treatments. It's something I've been really interested in, especially on the drug development side.
Yeah, you mentioned that before, so let's break it down. iPSCs, it's like hitting the rewind button on adult cells, right?
Exactly. Skin cells, blood cells, you name it. We can take these adult cells and reprogram them.
Reprogram them to do what?
We're basically turning back the clock, making them behave like embryonic stem cells again.
Wow. Okay, so they become like stem cells again. Why is that a big deal?
It's a big deal because now these cells, they can become any cell in the human body. Any cell.
Hold on, so we could turn a skin cell into a heart cell, a lung cell.
Yep. Even the neurons in our brains, that's where it gets really interesting for neurological diseases.
So scientists can create neurons from a patient's own cells. That's amazing.
It is, and that's the game changer I was talking about. We can start thinking about personalized medicine, treatments tailored to your genes. Imagine testing drug combinations on your own neurons.
No more one size fits all. That's incredible.
Right, it's huge.
Okay, but how do they actually do it? How do you turn an iPSC into a brain cell?
It's a process called differentiation. We expose the iPSCs to certain molecules, growth factors. It's all carefully controlled.
So like a recipe.
Yeah, kind of. Each ingredient guides those stem cells to become the type of brain cell we need. Neurons, astrocytes, you name it.
And then researchers can use these cells, these brain cells, to like recreate diseases in a dish.
Exactly. We call them disease models. Alzheimer's, Parkinson's, even rare genetic disorders. We can study these diseases in ways we never could before.
It's amazing, right? Instead of relying on mice who, let's face it, their brains aren't exactly like ours, now we can actually study these complex brain diseases in human cells.
It really is groundbreaking. We have a window into the human brain, and we can study these diseases at a level of detail we could only dream of before. Plus, you can make sure that the cells reflect a patient's unique genes.
It's incredible. And we've talked about understanding these diseases better, but what about treating them? What can iPSCs do there?
The potential is huge, honestly. One of the most exciting things is drug screening.
Drug screening.
Imagine testing thousands of drugs, potential drugs, on these iPSC-derived cells.
To see if they work against a certain disease.
Exactly. Like, we could test drugs specifically on motor neurons created from ALS patients.
Oh, like that study I read about, where they took these motor neurons from ALS patients and they tested already existing drugs.
That's the one. It's called drug repurposing, and it's a really hot area of research right now.
And what happened with that study?
They actually found something really promising. Digoxin. It's a heart medication, but it showed potential in reducing that TDP-43 protein from clumping together.
Which is a big deal in ALS.
Huge. This could really change the game for getting treatments to people faster, too. Imagine a drug we already use for something else might help treat ALS.
Like finding a hidden treasure in your attic. But there must be challenges, right? With iPSC research, it can't be that easy.
Of course not. One of the big ones is making sure these iPSC brain cells, they're really high quality and mature. They need to be just like the cells in a real human brain, or our models won't be accurate.
It's not just about making a neuron, it's about making the right neuron at the right stage of development.
Exactly. And it gets trickier with diseases that show up later in life, like Alzheimer's.
Right, because those iPSCs, they're like brand new cells again. So how do you make a cell age in a petri dish?
Good question. Researchers are working on techniques for that right now. They can expose the cells to different kinds of stress, or they can change certain genes that are involved in aging.
Wow, it seems like we're on the verge of something really big with all this. But are there like ethical concerns with all this cell and gene manipulation?
Yeah.
It's a lot to think about. But even beyond the ethics, with all this research, all these cells, there's got to be so much data.
Oh, absolutely. It's a data deluge for sure. We're talking about analyzing cells at a microscopic level, their genes, proteins. It's massive.
So how do they even make sense of it all?
That's where things like omics and bioinformatics come in. These tools help us analyze these huge data sets, find patterns we might miss otherwise.
Like a high-powered telescope, but instead of stars, It's genes and proteins.
Exactly. And those insights can lead to new therapies, better understanding. It's all connected.
It really is amazing. This whole iPSC thing, it's blown my mind. And before we wrap up, I have to come back to that question you asked earlier
Which one?
about the mini brain. If we could make one from our own cells, would we want to know if it showed we were predisposed to a disease, even if there's no cure yet?
Ah, yeah. That's a big one. I think it speaks to the responsibility we have as we move forward with this research. Knowledge is power, but we have to think about the implications.
It's tricky. Knowing bitter could empower people, help them make choices about their health.
Absolutely. Early detection is huge. They could change their lifestyle, maybe even enroll in a clinical trial.
But on the other hand, finding out you might get a disease that has no cure, that's got to be tough. And then there's the worry about discrimination, people treating you differently.
Right. It's a sensitive issue.
Yeah.
It's crucial that we talk about these things, scientists, ethicists, everyone. We need to decide how to use this technology responsibly.
This has been an incredible deep dive, really eye-opening.
It's a fascinating field, and it's constantly changing, giving hope to so many people facing these diseases.
Thanks for joining us today, everyone. And until next time, stay curious.